Dear friends, welcome to Biology Made Easy. Today we will discuss about Griffith experiment. What is this Griffith experiment? It is one of the experiments to prove which is the genetic material. During 1869, Frederick Miescher discovered nuclei from the post cells, and ultimately Altman found that this is present inside the nucleus and is acidic in nature, so he gave the name nucleic acid. But what is the function of this nucleic acid was not known. During the same time, Gregor Mendel, in his famous hybridization experiments of garden pea Pisum sativum, he told that characters are transferred from generation to generation vertically, and where these characters lies, he suggested units or factors which carry this genetic material from generation to generation. Ultimately, Sutton, Boveri, Morgan, they concluded that the genetic material lies in the chromosomes, and they proposed the chromosome as hereditary material. But what is the genetic material? It remained elusive for a long time till 1928, when Griffith put forth his experiment taking Streptococcus bacteria and mice. This is a very famous experiment. So Griffith took Streptococcus bacteria, Streptococcus pneumoniae. Previously it was known as Diplodocus pneumoniae. Now this bacteria causes pneumonia, and it has two forms. One is a smooth form, around which there will be a capsule. It is S type. The other is rough, and this rough cannot cause disease. It is avirulent, while the S type is virulent. In S type, again there are different strains, S1, S2, S3, S4, like that. Similarly, in the R strain, there are R1, R2, R3, all these things are there. But for his experimental purpose, he took S3 type and R2 type, important for examination point of view. Then he conducted series of experiments, which now we'll discuss. So in the first experiment, what he did, the R2 live form of a bacteria was injected to mice, and it was found that the mice were quite healthy. There is no symptom of any disease. Then he gave S3 live form, and when it was injected to mice, the mice were killed due to pneumonia. Third experiment, when the S3 was killed and injected to mice, then they were again healthy. There is no symptom of disease. In the fourth set of experiment, he combined the first and third one, the R2 live form and S3 killed form, heat killed form. They were injected to mice, and it was found that the mice were all killed, they died. So this is quite unnatural death, because when the live form of R2 is given, they remained healthy. When the heat kill form was given, they were healthy. But when both are combined together and the mixture was injected, all the mice were killed. So whenever there is natural death, you know that doctors do the post-mortem. So here also the post-mortem of this mice was done, and it was found that the mice were killed due to pneumonia, and their body, the live form of S3 bacteria were there. It's quite surprising, because we have given R2 life form, S3 heat kill form, but the bacteria died due to pneumonia, and again in its body there is S3 life form. How is it possible? So Griffith suggested something has transformed the R2 type into S3 type. Something. And what is that something? He suggested that there is some transforming principle which has caused this. But what is the chemical nature of this transforming principle? He put into ascertain, and he suggested it might be a protein. But ultimately, what is this transforming principle? It was solved after again 15-16 years in 1944 by three scientists, Avery, MacLeod and McCarthy. So these three scientists, they repeated the experiment of Griffith, but with slight modifications. They used three types of enzymes: protease, which degenerates proteins; RNase, which destroys RNA; and DNase enzyme, which destroys the DNA. And how they came to the conclusion, now we mark. Now this is the R2 form, and this is the S3 heat kill form. Now this is S3 heat kill form was given with no enzyme, and it was found that within the culture medium there is a S3 type of bacteria when there is no enzyme. Second, in another set of experiment, this S3 heat kill form was treated with the protease enzyme. That means the proteins will be degenerated. And here it was found that here there is again S3 type of Streptococcus pneumoniae. Third, in the third set, the enzyme RNase was given. That means the heat kill form there is no RNA. And again they observed that here is again S3 type of bacteria there. In the fourth experiment, they treated it with a DNase enzyme, and here they found there is no bacteria. So simply you observe it, you can come to the conclusion. When there is no enzyme, there is a S3. That means this heat kill form is transforming the R2 into S3 type. When there is no protein, but RNA and DNA are there, again the same thing is happening, transformation is taking place, that means proteins have no role. Again, when it was treated with the RNA, that means in the heat kill form there is no RNA, but protein and DNA are there. Then again the R2 is converted to S3. In the final experiment, when it was treated with the DNase, that means the heat kill form there is no DNA, but RNA and proteins are there. Then there is no transformation, there is no bacteria. So which is the transforming principle? Now it is concluded that the DNA is the genetic material. It transforms the R2 type into S3 type. So the genetic material is DNA. It was first concluded by Avery, MacLeod and McCarthy. But here one important thing, most of the students, they confuse how actually this transformation is taking place. I am explaining it very briefly. Now you see, suppose this is a tray where this is R2 life form and this is S3, but it is heat killed. Now here there is DNA. And here also there is a DNA, but this is smooth type, this is rough type. Now when this S3 type is heat killed, the plasma membrane becomes dead. DNA is a macromolecule. Previously it was unable to come through the cell membrane, but now this DNA can come outside to the medium. Now from this medium, if somehow it will enter into the R2 type, mark, I am using the word somehow. If somehow it will enter, now it will synthesize a capsule around it, and this R2 will be converted to S3. Why I use the word somehow? Here it is heat killed, so the cell membrane is dead, but this is living. The cell membrane is not dead, and DNA is a macromolecule. It cannot enter easily. That means very rarely it may enter. That is why in nature, transformation is a rare experiment. Only in 5% of bacteria, transformation takes place. In other bacteria, mostly conjugation and transduction takes place. This is very important. So, from this Griffith experiment followed by Avery McCarthy, now it is confirmed that DNA is the genetic material. But at that time, scientists didn't believe, because the amount of this nucleic acids, DNA, they are very small in the cell, while proteins are abundantly present. So how can it be DNA? It must be protein. They didn't believe it. So ultimately, the second experiment is that of Hershey and Chase, which unequivocally proved that DNA is genetic material. But that we'll discuss in the next video. Now we'll discuss some questions. So, number one question. Which of the following were used in Griffith experiment? A choice S3, R3, then B choice S3, R2, C choice S2, R3, D choice S2, R2. Now come to the second question. Use of which protein proved that DNA is genetic material? A choice protease, B choice RNase, C choice DNase, and D choice all the enzymes. Now come to the third question. When dead R2 type and live S3 bacteria were injected to mice, what will happen? A choice, it will remain healthy, it will suffer from pneumonia. Number 3, it will have no effect. D choice, anything may happen, cannot be predicted, anything may happen. So after going through the video, you will be able to answer all these questions. Thank you very much for watching it. Have a good day.